Well, welcome to our GRG online meeting. I'm Johnny Adams. Today is Saturday, June 1st, 2024. And our speaker today is Tom Benson. Tom's the founder of Mitrix. And Mitrix and Tom are doing some really exciting things in biological age reversal and with mitochondria. So having said that, short and sweet, Tom, I'll turn it over to you.
Well, thank you. I appreciate it. And thank you for that professional introduction. So yeah, I'm happy to be here. And I was going to give you our introduction to what we're doing and talking a little about some of the things that have happened recently as well. There's some people here who actually I've worked with quite a bit already, and there's a bunch of new people. So for those of you who've already seen this, you could, just listen to the parts you want. Let me start by bringing up a screen here. All righty, everybody can see the screen.
I can.
Okay. So, yes, what we are aiming to do is, and what we are doing is reversing age and also potentially curing a great number of diseases using bioreactor-grown mitochondria that are transplanted into the body. That sounds a little bit like science fiction. It's new, but it's actually, it is a very, very hot field right now. It's research going on at universities all over the world. It's about a 10-year-old concept. First developed some time ago. And it is really gaining speed because it has a lot of potential benefit medically. So let me just explain to you, first of all, I'll just want to say this is pretty experimental stuff. We typically, when we're working with volunteers and people, we typically work with people who are technically savvy. But please don't go out and try to do this on yourself, because it is scientifically complex. I also want to say we have a very, very good team. We've been around for about 3.5 years now. We've got some of the best scientists in the world. A couple of people from Stanford and UCLA as our advisors, who you probably recognize. Tom Rando is very well known as an aging expert. And then we have people from various universities, all of whom really are mitochondrial experts in their own right. And what happened was that a few years ago, I basically started contacting these people and said, "Hey, let's put together a North American team to go after this." So, and I also want to say that the goal, which is now being expressed in this new project that we just launched a few weeks ago, the big goal is to be able to do full age reversal of the body. Somebody is not muted. Yeah. Okay, good. Thank you.
Everyone mute your microphone, please. I was muted already, so I'll mute again.
We're aiming eventually, and very soon now, we're aiming to do to get people to 130-year lifespan. That's the big goal. And we have a team now of 15 or 16 volunteers, most of whom are in their 70s and 80s, who want to be part of this expedition. Okay. But let me back up now and tell you where this comes from. So the analogy we always use is, let's say that, you're looking at, this is actually my Toyota minivan. It's actually much uglier than this now. That's a few years ago. It's the paint's peeling and so forth. And if anybody's ever had one of these, they know the engine just never dies. It keeps going and going and going. We've had this thing 25 years. The kids have smashed into things with their learning to drive, and it's been taken on 1,000 vacations. But the one thing that I learned years ago was that every 4 years, the battery goes bad. Okay. And so you have to take it to the shop and give them their $200 and they pop out the old battery and they put in a new battery. Okay. And then you get more. So our philosophy with mitochondria is, what if we could replace your mitochondria the way that you replace the battery in a car? Which again sounds like science fiction, but as it turns out, based on this work that we've been doing, has been done by many researchers all over the world, the research is showing that in fact mitochondria are extremely replaceable, as it turns out. They don't just sit in the cells and just sit inside the cells. Mitochondria are constantly transferring around your body. They're being moved around. They're transferring themselves. They go between cells, actually go through the walls of the cell. They go through most of the membranes that we've ever tested in the body. They are carried by extracellular vesicles in your blood. Every one of you right now has almost 1 trillion mitochondria in your bloodstream contained in the platelets. Okay, you have 100 billion platelets. Actually, you have more than 100 billion. You have, oh no, you have 1 trillion platelets. Every one of your platelets has 5 mitochondria in it. And when that platelet gets to the end of its lifespan, it donates those platelets to other tissues. So your bloodstream right now is absolutely packed with mitochondria being transplanted. By the way, for people who forgot what a mitochondria is, the mitochondria are the little organelles that you have inside each of your cells. They generate all the energy. Without them, if your mitochondria were to just all stop right now, you'd be a puddle of goo on the floor. There is no life without mitochondria. All of your brain, all of your thoughts are being maintained by mitochondria that are constantly working and generating energy. Okay, everything that happens in your cell is based on those mitochondria. You have about 100 per cell, and you have 5 mitochondrial DNA per mitochondria. And I'm about to show you what a mitochondrial DNA looks like. But just so you know, the point of all this is that as we age, according to all the research we're looking at, our mitochondria decline. We start with perfect mitochondria when we're an egg, and it's all downhill until we get to be 85, 90, 95 years old. And then the mitochondria give out, assuming we haven't gone and gotten ourselves killed in some other way. The mitochondria give out, and we start to we run out of energy. Cells can't do their jobs. And there's all these age-related diseases that crop up even before that based on the loss of mitochondrial energy. And we'll talk a little bit about this. Most age-related diseases now are either caused by or they have some influence from the mitochondrial decline. Okay, that is what most researchers believe. And I want to repeat again, all of this is new. It's very theoretical. This is the work that we've done. You're going to find lots of people out there in the world, including other mitochondrial experts, who will disagree with many of these things that I'm going to say. This is our working hypothesis, which has been quite good. It's been very successful. So we're pretty happy with it. So this is a picture of mitochondrial DNA. It's actually a picture of about 500 strands of mitochondrial DNA that we got from a urine sample. This actually happens to be from my mother, okay, who is now 95 and in great shape, and was kind enough to donate for her son's crazy project. And each strand of mitochondria is one of these gray lines here. So you can see there's a whole bunch of gray lines. The white areas are just areas where the machinery lost a segment, so don't worry about those. This is all done with DNA analysis. So what we do is we stack up a whole bunch of the mitochondria that we get from the sample so that we can do statistical analysis on it. The black lines and the little gray and the little blue dots here represent damage to the mitochondrial DNA. And you can see here, and we have an algorithm. So we've figured there's about 25% damage. If you look at this one, this is from a 62-year-old donor, which happens to be me. This is my mitochondrial DNA, okay? I have exactly the same mitochondrial DNA as my mom because it is passed from mother to child with no changes. This is completely different from nuclear DNA, which everybody thinks about DNA as the nuclear DNA that you get from your mother and your father, and it combines and it's got 23 chromosomes. Most people don't realize you have an enormous amount of mitochondrial DNA. It comes straight from your mother, and it came from her mother and her mother and her mother. So it's really a family DNA. If you go to a fifth cousin once removed, you'll find exactly the same code. Okay, so it's very interesting. And that traces all the way back to mitochondrial Eve in Africa 300,000 years ago. So if you look at the two mitochondria, you notice the difference. Look at how trashed my mom's mitochondria are, and mine are less so. This is my nephew, who's 32, who has the same mitochondria as I do because it came through my sister. His are even less damaged. He has a damage score of 7%. I have 11. So we can chart these damage scores, and we can see the decline of mitochondrial quality over the course of the years. And by the way, this is not exact at all. I mean, we started at 60%. This is a very new field, the analysis of mitochondrial DNA. There are people who are doing this on a very large scale, this kind of analysis, and finding all kinds of amazing correlation between mitochondrial change in the body and disease. So if your curve is this blue one, then, we figure, oh, well, you're probably going to make it to about 95. If your curve is this gray one, you're probably going to die younger. So a lot of what we see as death from old age is really controlled by how quickly your mitochondria are getting burned up. Or perhaps you started with a bad set from birth. There are people that are born with bad mitochondria in some percentage. If there are children that are born with a horrible disease called primary mitochondrial disease, they don't even live, in many cases, they don't live past the age of 2. It's terrible. And because their mitochondria they're born with broken mitochondria from birth. As I always say, they're born with 100-year-old mitochondria in a 6-month-old body, okay, which is a terrible thing. But if you're a smoker, you can almost be guaranteed that your mitochondria are going to be getting burned out really rapidly. It was one of the reasons that smokers die young. If you get chemotherapy, you'll actually see a big drop, like boom, because chemotherapy kills mitochondria. That's what it does. It's actually designed to do that. If you get any chronic disease, we're actually looking at the possibility that long COVID may be caused to some extent by permanent damage to the mitochondria in the body. And then there's also this question of what happens if you get below a certain point. We think that what happens is it triggers a whole bunch of these age-related diseases because the body's struggling to deal with the lack of energy and these other things, for instance, Alzheimer's. I just had a meeting with 2 Alzheimer's experts yesterday, and they're absolutely convinced that Alzheimer's is triggered by mitochondrial deficit. The cells don't have enough energy. They can't keep the waste products, keep going with their waste disposal, which is very important in the brain. The things get mucked up, and that causes Alzheimer's, okay? So here's the point of all this. What we want to do is give you a boost, okay? And you can see here where I've very hopefully and optimistically drawn this line out to 130, 140 years old, okay? If we could boost your mitochondria every whatever number of years to make up for the fact that they're continuing to decline, maybe we can keep people older, keep people going. And we are doing this with animals, and it works. We're doing it with animals a lot. And there's a lot of other people too. It's not just us. As I said, there's teams all over the world right now working on this. So let me explain how this actually works. When we talk about mitochondrial transplantation, it really, to you, it would seem as simple as a blood transfusion, okay? We've even looked at the possibility of injecting it. You might even be able to inject it yourself. People get therapies right now where they inject immune components to bolster their immune systems. And a lot of this now is being done at home. But you would essentially would look like a bag of yellow liquid, and you would put it into your blood, or maybe you inject it into your tummy fat. And what happens is that the mitochondria in that solution, there's a lot going on behind the scenes. There's a huge amount of biology and chemistry that goes into making this work. But to you as a patient, it would just look like a blood transfusion. By the way, I just want to mention again that mitochondrial transplantation within the body is what, as they say, evolutionarily conserved and pervasive. It's happening everywhere. When you get a stem cell therapy, a very big part of the value of the stem cells is the fact that they donate freshly made mitochondria to the tissues nearby. They actually extrude these little tubes. And we have pictures of this. We have videos. You can see the mitochondria marching down this little tiny tube and to the cell, okay? And they don't do it with cells. They don't do it with cells that are healthy. They only do it with cells that are old. So there's a signaling mechanism in the body where the cells can actually say, hey, I need more. And the stem cell comes over and gives them a replenishment, okay? So when we talk about doing mitochondrial transplantation, we're just doing what the body's already doing. You already have in your bloodstream a thing called mitlets, what we call mitlets. Mitlets are tiny, as I think I mentioned to you, your platelets have, your platelets in your bloodstream contain mitochondria that are fresh and young. They were manufactured in your bone marrow, given to the platelet. The platelet lives for about 10 days. At the end of the 10 days, it's taken off to be recycled in the spleen. But right before it gets recycled, it squirts out all of its mitochondria in these little lifeboats, which we call like extracellular vesicles or mitlets. Okay, I actually made up that name. And they get absorbed by all the other tissues nearby. And so mitochondria are recycled. They're never thrown away by the body if it can possibly avoid throwing them away. They're not easy to get, and they're not easy to get from nature because they're so valuable. They're super valuable. They're hard to make. They require a tremendous amount of energy to build and a tremendous amount of energy to maintain. So your cells are very, very well evolved to keep your mitochondria healthy and to recycle them constantly, okay? And that's why we live to be 85 years old instead of, some animals only live to be 30. We live to be 85. One of the reasons is that we have very good mitochondrial recycling in our body. And here's a little picture of a nice little cartoon of the platelet releasing the mitlet with the mitochondria inside of it. It floats over, it gets absorbed by neutrophils, it gets absorbed by white blood cells, T cells, and re-energizes the immune system. Here's another graph from a paper where they showed the flux of mitochondria in the blood, and you can see it's going down, okay? And also the mitochondria are less powerful. As I said, the mitochondrial DNA tends to decline. And so that means that the DNA is no longer functioning properly, which means that the mitochondria is no longer generating energy properly because it's got bad code in its DNA. And we think that that is one of the 2 or 3 top causes of aging. I know there's 15 hallmarks of aging. We're trying to look for what are the actual cause as opposed to the downstream effect. We think that mitochondrial DNA degeneration and nuclear DNA degeneration, which was what people call epigenetic damage, those are the 2 core reasons that we age. It's genetic decline, okay? Some of you are probably following the epigenetic reprogramming field, and there's a bunch of companies working on that. There's a lot of very, very well-funded organizations who are working on epigenetic reprogramming. From our perspective, we want to team up with them because we're doing the mitochondria side, they're doing the nuclear side. Both of those ideally should be repaired in order to do age extension, life extension. So the next big question is, really the main question is, okay, great, mitochondrial transplantation. Where are we going to get them from? Okay, we can't squeeze them out of teenagers. The teenagers are not going to sit still for that, I guarantee you. Okay, and it's not like we need small amounts. You need vast quantities. You are losing, according to our best rough estimates, by the time you're 80, you're losing somewhere between 20 and 40% of your inventory of mitochondrial DNA. That's quintillions of mitochondria, enormous numbers, okay? You are 10% mitochondria by weight, okay? So we're not talking about small numbers here. We have to, if we're going to refresh you and refill you back up with new mitochondria, we're talking about quadrillions and quadrillions of mitochondria. Where are we going to get them all? Well, the only way to do this on any kind of socially acceptable, ethical way is we have to grow them. And so that is what our primary focus is as a company. Here's another little diagram that kind of shows the whole cycle. This is one way of doing this. We take the mitochondria from your stem cells. We take some of your stem cells out of your blood. We put them in great, big, enormous 2,000-liter stem cell bioreactors. We grow vast amounts of it. We extract the mitochondria from it. We put it into some coatings to protect it from the immune system, and then we inject it back into your bloodstream. And the coating would have, ideally, the coating will have a receptor built into it so that we can actually target them to specific organs. So if you've got heart trouble, or if you have premature blindness, you're getting AMD. I mean, by the time you reach 70, 80 years old, there's a lot of potential things that could be going wrong. If you get sick and you've got an infection, you're in the hospital, we want to be able to give your doctor a toolkit of mitochondria so they can fix whatever's, the biggest risk for you right at that moment. Okay. Otherwise, we just give them to you in your whole system, and your whole system just starts regaining strength. I'll just show you, here's some of the best data. We've been doing studies now for 3 years. We've done a lot of injections. We've looked at the brain, we've looked at the immune system, we've looked at muscle strength. I have a huge series of injections going on right now at one of our labs at University of Kentucky, where we've actually gotten up to the point where we're now injecting, I think we've hit 6% of the animal's mitochondria that we have supplemented now, and we've increased it 6% with young mitochondria into an older animal. This is what the kind of results we see when we do that. Okay, this was an experiment where we took older mice, the equivalent of maybe 60 years old by human age, and we gave them a fatal dose of sepsis. The orange line are the mice that got that sepsis and did not get any kind of a treatment. Within 24 hours, they were all dead, bacterial infection. The blue line is when we gave them Mitlet injections. We gave them 3 injections right at the very start. And you can see 1, 2, 3. And then they slowly, they still died, but we slowed it way down. Okay. We think we're going to go back, redo this, and give them and continue giving them injections. We think that after a week or so that they'd be cleared completely of the disease. We tested their cytokines, we tested their blood bacteria, everything is down a factor of 10. Okay. We also did this for viral infections, the H1N1 flu, same thing. We have another person who did this at Stanford, and they tested this with T cells, specifically designed to do this with T cells. And I think they were testing it against TB, tuberculosis virus. And this was in a test tube, not in a mouse. I got the same thing with T cells. So if we can boost T cells, then you begin to think, well, T cells go against cancer. Okay, so we really want to try this against cancer. Maybe merge it with CAR T therapy. So you can see there's all kinds of opportunities here. Just looking at the immune system, if we can supply spare battery packs, think of your immune cells as being little roving battery powered units. The mitochondria, when they're out there fighting against an infection, their mitochondria get burned out very quickly, just like batteries. And we want to do is give them spares, and that's what we did here. We gave them this big chunk of young, healthy mitochondria. And so, from a human perspective, if you're 75 years old, you're in the ICU with some disease, they can't get rid of it. Maybe we could give you a big old dollop of young mitochondria and make your immune system think it's 30 years old again. That you get out of the hospital, because young people with young immune systems just don't get as sick. So the opportunities are pretty big. I'll skip over a lot of this other stuff. We're doing a bunch of tests in doing eyes, doing vision correction. You can inject mitochondria directly into the vitreous of the eye, and they actually migrate into this picture over here, this kind of weird looking thing. That red up there, that's a glob of mitochondria in the vitreal fluid. This is what they call the optic nerve head. And you can see this red color. Those are the mitochondria. You can see them. They're actually floating down and getting into the optic nerve head, and they're migrating into the optic nerve. Now, if you can replace the mitochondria in the optic nerve, then you can potentially prevent glaucoma, because glaucoma is a mitochondrial deficiency disease. So I'll jump over the rest of this. So if you talk about, and as I said, a lot of people doing this work. And if you go to our website, we have tons of papers on this. So what's our goal? Our goal is to, obviously we'd like to cure a bunch of diseases, but the big goal is, can we use this technique to give ourselves longer lifespans? And yes, we would be healthier, we would be younger, we'd have stronger muscles, but it isn't just about, I want to get past that 95-year mark that seems to be the limit. I want to get to 120. I want to get to 130, okay? I don't think the mitochondria are the entire solution, but the approach we use is what we call a laptop refurbishment model. We figure that to refurbish our bodies, we're going to need 3 or 4 different treatments. And if anybody who's ever rebuilt the laptop can tell you, the first thing you do is you put in a new battery. Because if you don't have the energy, you can't rewrite the operating system. You can't do any of these other things that you need to do. You can't clean up the hard disk. You need a fresh battery. That's just minimum requirement, okay? And so that's our goal, is to replace the battery, okay? And then there's other people who could do the other pieces. I better look at some of my chat questions here. Okay, someone said, Tom, what is the conceptual link between your work and the life extension PQQ product? I don't know what that is, but I'm assuming you're talking about, there are a variety of vitamins and supplements that are purported to help mitochondria. There's also red light therapy, which is using LEDs to put extra energy into the mitochondria. There's a bunch of companies actually developing some pretty serious, what I call chemical enhancements. So the difference between them and us is just a difference of how aggressive we're being. Think of what we're doing as being 10,000 times more ambitious than what they're doing. I mean, yes, you can certainly improve your diet and you can make your mitochondria a little better. But the bottom line is, going back to our picture way back here, here's the decline of the mitochondria, okay? If you're down here and you're 90 years old and your mitochondria have declined 40, 50%, sure, if you improve your you improve your diet, you might get 1% improvement. But then it's just going to keep going down because this decline is based on genetic damage. It's actual noise that's creeping into your genes, into your genetic structure. You can't fix that with a nutrient, okay? The nutrient helps the mitochondria work a little better. It might supply some chemicals it needs, but you can't change the fact that you've got this genetic decline. The only way to fix genetic decline that we know of is to just give you new ones. Okay. And going back to the battery analogy, I remember when I was a kid, you guys probably remember this, they used to have battery rebuild kits where you could, if you had an old battery in the car, you could get, you could pour new fluid in there and you could try to put in chemicals. And they said, oh, yeah, it'll keep your battery going longer. We don't do that anymore. All the batteries, they're sealed. You can't put new fluid in them. You can't put nobody sells battery extenders because it just doesn't really work. Once the battery goes bad, the electrodes get worn out, the chemical goes bad. Easiest thing to do is you just pull it out and you put in a new one. And that is the way all cars work these days. And so that's what we're saying. We're not trying to make the mitochondria work better. We're just going to give you new ones. Okay. So I don't take them myself. I make sure I eat my vegetables. Okay, Walter had a question. Can normal distribution of mitlets that happen to be diseased be a cause of spreading pathology? That's a good question. I don't know. Probably. Once again, keep in mind, we're like a teeny little company. We don't even have an office. I have all these labs working for us, and I'm running this all out of my home office. So this is really new, and there's so much work to do. I mean, we could spend 50 years working on this in the world and take a long time to uncover it all. Somebody said, I'm 89, so my mitochondria probably have significant genetic damage. Yes. If you clone them, doesn't that damage just gets passed along? Thank you. That was exactly the question that I should be addressing next. Let me go back to the bioreactor thing. So let's say we put your stem cells into a bioreactor and we expand them. And by the way, generally what we're saying is we're going to expand your stem cells about 10,000x. That's how much we're going to do. It's a huge amount. Well, if we're just got 89-year-old mitochondria and we inject them back into you, that's not going to do any good. You already have those. Our process is set up to eliminate the bad ones during the growth process. And I don't talk about exactly how we do that because that's secret. But if you think about it, if there wasn't a way to make mitochondria younger, our species wouldn't exist. There'd be no babies, because the bottom line is every human egg cell contains somewhere between 300,000 and 500,000 copies of the mother's mitochondrial DNA. People don't know this. Everybody thinks you got an egg cell and you've got a 23 chromosomes in there from the nuclear DNA and it gets fertilized and it creates a new DNA, right? And that's about 8 billion. It's about 8 billion base pairs that are in your nuclear DNA. People don't realize that your egg cell has almost exactly the same amount of genetic information, somewhere between 6 and 8 billion mitochondrial DNA. 500,000 copies times 16,000 bits, 16,000 base pairs is around 6 to 8 billion mitochondria. So your egg cell actually contains a completely alien DNA. Okay, it's not yours. It's passed down from the family. And they never talk about this in the textbooks because it wasn't really even known up until a few years ago. So that perfect mitochondrial DNA is the reason that babies are young. That's what youth is. And how does the female reproductive system create those egg cells with that perfect mitochondria when her mitochondria are damaged? Of course, the older she gets, the harder it is, right? Because she's got less and less healthy mitochondria to get. But we know that there is a technique that the female reproductive system uses to clean out the bad ones. However, it is extremely energy intensive, which is why our bodies don't do it. If there was a simple way to clean up our mitochondria and get rid of the bad ones, we'd already be living to be 300 years old. Okay? The reason that we age is because it takes more energy than our bodies can maintain. That's my theory of aging right there. Animals that only live to be 30 years old, like antelopes and wolves, and so forth, we think that one of the reasons is that they burn their mitochondria out faster because they have to run. They've got to escape. We're primates. We don't run around and use energy at those levels. We come from a primate line that is optimized for age in order to optimize for the knowledge of the elders, so that the tribe, if you look at a group of gorillas live to be about 60, okay? They're very, very long-lived for their size. The reason that gorillas have that long life is because they're protecting their mitochondria and they're protecting their elders, because the elders have the information that the group needs to successfully live in that area, right? And the human race is especially that way. We're very good at extending our mitochondria. This is all theory. So the bottom line is that we have to grow the mitochondria in enormous quantities, and then we have to, at the same time, we have to cull out the bad ones. So this is going to take a while, and it will take a lot of energy and a lot of materials. This is going to be a huge tank that is basically cloning your cells and improving them along the way. That some people have also said, could you just make these? Could you just, since this is all family mitochondria, couldn't you just use a generic group of mitochondria? And that is possible. I mean, if you go to Sweden, for example, the truth is, if you go to these countries where there's a very heterogeneous ethnic group, most countries actually, other than the United States, we're kind of a mix of everything, right? But if you go to Sweden, if you go to Ethiopia, people generally have very, very almost identical mitochondria. So is it possible that the Swedish government could grow a great big giant factory of mitochondria and just distribute them to everybody? Sure. That's absolutely a goal. The long-term goal is to manufacture these on a global scale and give them as a supplement to everybody. But as you can imagine, that's a big project. Martin asked, are we epigenetically reprogramming the mitochondria? No, it's a different thing. We are doing the equivalent of that. We're just getting rid of the bad ones. We're not actually reprogramming them. Other people are trying to do that, however. There's other companies that are trying to epigenetically reprogram the mitochondria. So it's not that it's not being tried. That's just not our approach. Epigenetic reprogramming in the nuclear DNA, of course, that's where you send in new code and you basically kind of paste it over the existing bad code, shall we say. It's like rewriting the operating system on your hard disk. So let me just see if there's any other interesting tidbits here. I mean, obviously, I'm not going to go into detail about our company. We've got patents and so forth here. You know, we've got a number of different approaches to this. We are raising money, by the way. I'll just mention that. But I want to go back to this first diagram where I talked about this process. And as I said, by the way, Martin, who's on the call here, is one of our volunteers. Thank you, Martin. And Elaine. I think Elaine's probably there too. I can't see her picture. And Walt is also a volunteer. So we've got a number of you here. We also have a number of people in your group who've invested. Fiona Miller is one of our investors and has been from day one almost, and has been a great supporter. So what this is all coming into here right now is this idea that we're going to actually try this, okay, in human beings. And I just want to mention real quickly, why are we doing this now? The first question I always get from other scientists is, they always say, well, God, shouldn't you wait another couple of years? Do this in mice more, learn more about it. That's certainly the way that most biotech works in the United States. You take your time and you go slow. What about doing this in dogs? What about doing it in monkeys first? And we've been through all that. Actually, we did this we did a dog trial and we got the same results. There's nothing in this that tells us that it's going to change when we go to different animals, except the immune system. Mice have very easy immune systems, so you don't have any kind of reactions. Human beings have extremely fussy immune systems. So immune reactions are a big concern, absolutely. Dogs' immune systems are not that reactive. So the bottom line is the reason we're doing this now, and I mean, our goal is to do this next year. Basically, as soon as we get funded, get the funding we need to get, we're gonna go. And of course, we would have already been doing this if we'd been able to get the funding a year and a half ago, but the funding market just collapsed. So it's recovering now. So I'll just mention, why are we doing it so quickly? Because you have to start with human beings eventually. And when you go into human beings, you're going to have to start from scratch. That's the bottom line. I mean, you have to do it so safely and so carefully and so slowly that doing it in animals is almost I don't know that it has a lot of value. And there's a lot of ethical issues. And the animals can't tell you very much. A lot of this it's very hard to judge how successful this is. And a lot of it's going to be whether it feels good to people. This should be the reverse of chemotherapy. When you get chemotherapy, you feel terrible because you're poisoning your body. This is really the complete opposite. We're trying to recharge you. And so a lot of the human element is important to us for these kinds of trials. And I also want to mention that these trials, this will be the first time we've ever done it at this scale in human beings. And we want to establish safety for the world. I mean, there's a lot of people who want to do this. This is a big industry. So our volunteers are going to be, really pioneering this. So let's see. Someone asked about volunteering. Just contact me if you're interested. And somebody also asked about cell sorters. We're not really using cell sorters are an idea, but we're not using them. So why don't I let's see, how much time do we have here? Oh, that was 50 minutes. That was 45 minutes. That's plenty. Why don't we just throw it open, and anybody who wants to chat or ask questions, please. Anybody else have
Well, I had learned somewhere that mitochondria have a lot of effects in managing and controlling things in the body.
Yeah.
And you've talked a lot about energy creation, but what about managing and controlling?
Well, that's a big question.
Mhm.
No, they're making enormous strides in understanding mitochondrial contribution to the management of the cell and the management of the body. And, basically, remember, the mitochondria have been part of us for 1.5 billion years, okay, or 1.25 billion years, whatever it is. So they were here before we were. We were built around them. That's what I will say. We were built around the mitochondria, okay? So that's why I say, when I say we're a robot they built so they could go and explore the world. They and the mitochondrial DNA and the nuclear DNA are in constant communication. In fact, there have been some researchers who've stated that mitochondria are actually where most of the processing is happening, because processing takes energy. So it actually makes sense that you'd have the processing happening in the mitochondrial DNA, and the nuclear DNA serves more as a database. But the actual day-to-day processing and the sensing. Those mitochondria are floating around all over your body and all over your cells, and they're collecting information inside the cell and passing that information back and forth to the nuclear DNA, so that together they can make decisions about what to do. Because running a cell is a fantastically complicated. We have no idea how it's all managed. It's out of this world. It really is. And then just running the mitochondrial energy production, it's got to deal with all kinds of changes. And what happens if you're in a famine? What happens if you're in low oxygen? What happens if you're eating a lot more fat than you are glucose? That mitochondria, it's got a tremendous job to manage its energy production. And don't forget that the surface of the mitochondria is covered with tiny little jet turbines. That's how it generates energy. It actually has a spinning molecule that is spinning at 5,000 RPM. And it's generating little ATP molecules, and it's driven by hot gas. It is a jet turbine. Okay, literally. And your life exists. You exist because you are full of tiny little jet turbines. And so, as Yoda would say, we are energy beings, right? Let me get up here. Okay, what's your contact information? Oh, I'll put it up here right now. It's tom@mitrix.bio.
I have a little question, if that's okay.
You can go to our website. Actually, the easiest thing is just go to our website and go to our contact page, and that'll get through to us. Okay. I'm sorry, go ahead. Next question.
I was going to ask, and this maybe it's more practical for you than anything else, but for people who are getting treatment, if we happen to have a matriarchal line of, for instance, my sister's grandchildren, my sister's granddaughter, I guess. Well, no, any of my sister's grandchildren could provide my mitochondria, right? And it would require less filtering. Does that have any value to you, or is your filtering
Maybe.
good enough?
Maybe.
Oh,
Maybe.
okay.
Sure. I mean, if one of your sister's daughters, decide to have a baby, boy, the placenta is a great source of young mitochondria. Okay.
Oh.
And I would save that. But again, we may figure out, oh, we don't need it. It's like, you remember when everybody was saving cord blood for the stem cells?
Yeah.
We did that when the kids were young, and then it's eventually they kind of figured out, we don't really need it because we know how to get it from your own body. But this is where placental stem cells are still being used very widely.
Yeah, I don't want to be known as Uncle Dracula anyways.
Uncle Dracula, exactly. I want your blood. I just want a little blood. I told my kids that, but for me, my kids do me no good because they didn't get my mitochondria, right? So I have to go to my nephew, and he's like, "No, you ain't getting any of my mitochondria, Uncle Tom." What are the qualifications and disqualifications for participation? It's mainly we're just looking for people who are generally over 65. And by the way, I'll just mention, people say, "Well, how come I can't do it if I'm 45?" Because if you're 45, your mitochondria are already in pretty good shape. That's the truth. I know that we'd all like to be 25, go from 45 to 25, but you're at 45, your mitochondria are in pretty good shape. And so I would need so many young mitochondria to flush out to give you really any kind of noticeable boost. Whereas someone who's 70, 80 years old, their mitochondria are in bad shape. So it's going to be really easy to see a benefit for them. It's just dilution factors. So we're starting with people, and this is really key to the research. We work with older animals. If you're working with young animals, you don't see anything. They've already got young mitochondria, so that you don't get any signal out of it. We're also, we're being careful we don't want to work with people who have active cancer, because cancer is a complex question. We think it's resolvable, but we don't want to deal with that now. And we can't work with people who have severe dementia. Just because we can't get, that wouldn't be, it's not legal. So there's a question here from Rick. What biologic and phenotypic markers will we be assessing for proof of concept? Well, just so you know, I mean, we've done this with animals, we've done this with, and so we get a lot of, we look at their muscle strength, we look at how much they can run on the wheel, we look at their, we look at the mitochondrial energy levels in their brain. If you look at our papers, you've seen we've covered everything we can do really. With human beings, what we'll be doing is looking at really, it'll be very similar to the X Prize that's currently being done. We'll be looking at muscle, cognition, immune system, and skin. Those are the 4 areas that we're going to be evaluating. A lot of this will be like with skin. I mean, you have to do a visual assessment. We have a judging panel of kind of prominent people, and the judging panel, part of the purpose of the judging panel is to make sure that this is all being done really well. We don't want it to be shady at all. This is where, I mean, we're working with Stanford and Harvard and some of the best universities in the world. So this is going to be very, very high-end project. And the judging panel will look, one of their jobs is to look at the skin and to look at the appearance of the body. And then you can do muscle strength. The trick is you can't just take someone and give them exercises and say, oh, they're stronger, therefore they're younger. We all know that's not real. It just means you're making your muscles stronger. It doesn't mean you've changed your age, okay? But that's really hard to separate out. And that's like a big debate right now in the field. So we have a whole bunch of external and internal. We have a lot of blood testing. We have our own test of the mitochondrial DNA that we'll be using, obviously, to guide to see whether or not we've improved the quality of the mitochondrial DNA. How much travel would the volunteers have to do? A lot, because this will be probably done in Japan or in the Bahamas. So there will be significant travel. Is there a test to accurately measure the health of the mitochondria? Yes. Is there a test to accurately measure the health of mitochondria? Yes. And that is the one I showed you at the beginning. Are we still sharing the screen, or did I turn it off?
It's off, but
It's off.
is that test available for anyone, just as a fee for
Not
service?
yet. We're not allowed to sell it because it's so new and we didn't get any approval for it. And so it's right now we're still just collecting. Part of what we'll be doing with all of our volunteers, we'll be doing tests on all of them constantly because we want to see the progression. Right now, that test costs many thousands of dollars to do because it's all being done manually. And so we can't just, and we can't sell it because you're not allowed to do that. So we can only do it for free. So we can only do it enough. It's kind of weird. You have to have the funding in order to do these things. Jason asked, are you able to create an exact match of a youthful mitochondrial copy of the mitochondria, only younger, or is it only from donors? No, Jason, the idea is that we could take see, everybody has a mixture of good and bad. When I say younger, what I really mean is that you have a lot of good, you have more good than bad. It's like black marbles and white marbles, okay? And as you get older, you have a larger and larger and larger ratio of black marbles to white marbles. The black ones are bad, the white ones are good, if that makes sense. And so what we're doing is basically cleaning out the black marbles so that you have more of the white ones left. What organs will you target, or full body? Full body is the main goal. We can target organs because we have a targeting molecule that we're building into the coating that we put on the mitochondria to protect them. They also have a targeting molecule that will take them over and put them into a specific organ. There's a lot of work that needs to go into that. We've developed a couple different versions of that for different organs. So can we use the VO Max test to test the health of mitochondria? Yes, that is going to be on our list for sure of things we test. But again, the VO test, you can tweak that just by exercising and riding your bike more. So we have to make sure that our people are already at the same level of conditioning that they're going to stay at, so that we know that whatever we're improving is not due to them just exercising more. Okay. And so our volunteers, we're going to be driving them to exercise. Most of our volunteers are already people who are really in excellent shape, who've already taken care of themselves. They've already been working on this. They're getting to be 78 years old, and they realize that, hey, I'm seeing this decline that I can't stop with any of the other techniques. And so they're all seeing it in their own body. Okay. Have you seen any side effects or adverse reactions? Not so far. In our animal tests, we've seen 0 adverse reactions. We have tried. We have not been able to get them. I want to mention this has been done in human beings, not just not by us. People have done this in human beings already on very, very small scales for very narrowly focused diseases. There's surgeons at Harvard who are already using these injections of mitochondria to restore the health of the heart during cardiac surgery. They bring the heart back to life because you always have some loss during surgery. And they inject mitochondria in there and they bring them back to life, and then they close up, and then you end up with more function. That's very common. They're already using it for treating clots, blood clots, and ischemia in the leg. They're using it to treat children who have mitochondrial diseases. There are some babies that it appears that their lives have been saved by mitochondrial transplants in Israel, at another company that's doing this in Israel. So this isn't the first time it'll be done for human beings. It's just we're doing it on this massive scale, and we're doing it to bring people back from old age as opposed to saving the lives of children. I also want to say our goal is, once we've proven this with older adults, we absolutely want to be using this to treat children, of course. I mean, we'll have a certain minimum amount of our project that we'll be doing pro bono for children's diseases. And again, this is a case where the adults are going to be testing this and making sure it's safe, so that we can use it for children. Other questions? I don't see any other questions. I've been talking and talking, so maybe that's enough. What do you think, Johnny? Is that enough, or should?
Any more questions?
Yeah.
Anyone?
I've got some more questions.
Go ahead,
So,
Jeff.
what is the protocol for the actual procedure? Is it one time, or is it repeated weekly, or how often do you get the treatment?
Very, very good question. And the answer is, nobody has ever done this anywhere ever. So the truth is, we don't know. Remember, this is not natural. It's never been done in nature before, that any animal has ever been able to refresh its mitochondria. So the answer is, I don't know. I mean, the current plan, at this point, we're guessing. We're figuring that we'll do a treatment, an infusion once or twice a month, maybe for a year. Okay. Maybe once every 2 months. Maybe we'll be able to do it all at once. I doubt it. Remember that we're talking about replenishing. We're talking about refilling you with anywhere from 2 to 10% of your mitochondria. So it's a lot. We're talking about adding massive amounts of new material to your body. Well, your body's going to need time, and mitochondria move around, and so it should work, right? And it works in the animals we've done it with. We have animals now that are up to 6% replaced mitochondria, and they are absolutely healthy. In fact, they're more than healthy. But we're not going to push them there. We've definitely overdosed them too, and that doesn't work. They die. Okay, you just overload somebody, and it's just too much. This is very similar to stem cell treatment, by the way. So for those of you who've gone and done stem cells in the Bahamas, or you've done them in Beverly Hills, you've gotten a facial, you've gotten knee injections, whatever you've done with stem cells, think of this as being stem cells times 10,000. Our treatment should be, according to our calculations, it should be the equivalent of 10,000 stem cell treatments. And that's how we get the effect. In theory, stem cell treatments actually are making you younger because they are donating mitochondria to your cells, but it's just too small. It's just not enough.
I got a follow-up for you, Tom. Quick follow-up on that question regarding like if you're thinking about the mitochondria, you were saying what you have, say white and black ones, white ones being the good ones. So what you're putting into the body, is this something that your body already has some of those? Like you may already have, I'm just giving like small numbers to make sense. If you have 10 white mitochondria already and you have say 10 black, you're really putting in 10 more white and trying to remove those other 10 black. You're not really putting in something from a donor or from something mysterious. It's really something that's already in your body. Is that right?
Well, look, there's, and yeah, I talked about this, but it's definitely complicated. So let me just refresh this. There's two approaches to any kind of regenerative medicine. You could do autologous or allogenic. Okay, those are the words. Autologous means that you're taking your own cells, your own mitochondria, and you're growing them and then putting them back in. Okay, it's your own stuff. Allogenic means you're taking somebody else's and injecting them into you. So if you go get stem cell therapy, there's two different types of stem cell therapy. There's the stem cell therapy where they take it from your tummy fat and they inject it into your hip. That's autologous stem cell therapy. There's also stem cell therapy where you get it from a donor. Maybe you get it from somebody's cord blood or you get it from somebody's placenta. That's allogenic stem cells. So, and the allogenic stem cells do not have your DNA in them. They have somebody else's DNA. So what we're talking about right now is autologous. We're going to grow your mitochondria, but we're just going to make sure that there's more of them that are good as opposed to bad. Right now, my mitochondria, let's say, are 70% good and 30% bad, which means 30% of my mitochondrial DNA have been damaged with some sort of bad code, some bad deletions in the code. Okay, it's like bad sectors on a hard disk. So I have 30% of my mitochondrial DNA, and they're still mine. They've been ground up and trashed by the day-to-day life. If I take my mitochondria out, that's 70-30, and I grow them in a bioreactor, and I figure out a way to reduce that ratio so it's now 90% good and 10% bad. You never get to 100%. But if I can get it to 90-10, and then I put them back into myself, I'm improving that 70-30 ratio.
Just a quick follow-up on that. I mean, say the 70% that are good also have some mutations in the mitochondrial DNA, even though that 30%, maybe it's like they have dramatic mutations, but even that other 70%, and I mean, I don't know if we can measure this or not, but say there's also, say those are still quite different than when you were 20. Are you able to recreate like a brand new mitochondria with the new DNA like it was when someone was 20, or would it still be using, you know what I'm saying?
I don't know yet.
Not sure yet. Okay.
The question, it's a great question. You are on the edge of even my speculation, much less my facts. Okay. So, I mean, we just don't even know. And as I said, every human egg cell starts with 300 to 500,000 copies of perfect mitochondria. Well, that's actually not actually true. Your egg cell has a mixture. And by the way, it's not true that they're all from your mother. There's actually sometimes other ones creep in. The mitochondria from their sperm sometimes float over and get in there, right? And so there's a huge biological, like species level questions coming up here about how does Mother Nature do this? And it is really interesting. And you could look at the research. If you want to look at the question of how youth is created, how do our bodies create a young, replicate of ourselves? You could read. There's a guy named Nick Lane out of the UK who has been studying this extensively. He's a mitochondrial expert. He gets quoted by Bill Gates. He's a great guy. And he writes lots of papers analyzing the energy development and the use of mitochondria in the gestation. So how is youth created? By nature. And we're trying to basically replicate that using a machine. We're creating a youth serum. I mean, it is a youth serum, is what it is. It's the same youth serum we already have in our bodies. We're just trying to make more of it.
Thanks, Tom. Appreciate
Sure.
it.
You mentioned that it is possible to overdose on this treatment.
Yeah.
I presume that was with animal studies.
Yeah.
How do you know what that level is with humans?
We're going to start really small. Okay. And we've done, that's what you do when you're, what we're doing with humans is it's called a phase 1 safety trial, right? And to do safety trials, what do? You do dosing studies. But we've done this with animals. We think that it should scale proportionally to weight. There's really no reason. I mean, the immune systems are different, so that is certainly a huge consideration. But we think that if you inject too many, it just, intravenous injection. If you inject too many stem cells, you can kill somebody. You just you end up clogging up the capillaries in the lungs. Lungs are the first place that any intravenous injection goes. So we had some mice. We gave them great big giant intravenous doses, and they died. So we did that until we figured out what was safe. We'll be doing a lot more of that before the human tests. That'll be one of the things we'll be doing, is a lot more safety tests in animals before we do any humans. And then when we do the humans, we'll be starting with really tiny, just skin injections, just to see if we see a reaction, and then we'll start with slightly larger ones and so forth. So, estimated future costs, high. It's going to cost a lot in the beginning. This is going to be, I mean, you can imagine, I'm trying to replace 10% of your mitochondria. That's hard work. But it's not really now costs. We just don't know. It's more about how much do we need to invest to build the infrastructure. Any technology. I mean, if I was the first person to make a semiconductor, how much would the very first integrated semiconductor chip, how much did that cost? Well, millions of dollars, many, many millions of dollars. But that's not the issue. The issue is, can you mass produce it? The question is, how much do we need to invest in capital to get up to mass production levels? And that's, a lot, millions and millions, hundreds of millions of dollars potentially, but that's just true of any mass production environment. So how much money did it take to build Tesla? Lots.
Well, outstanding. Any more questions?
That was more. That's an hour and 10 minutes of just talking. So any other questions?
Well worth the investment in time. More questions?
Well, great crowd. I mean, the nice thing is I'm getting people who are already thinking about this, and that is really wonderful. And a lot of you have already done your own tests, who've tried things. I hear from people all the time who've tried different interventions themselves. So,
well, that's what we're about here. Action and results, not just theory.
Right.
So, okay.
Good. Well, anybody, as I said, go to my contact page, leave me a note if you want. And thank you for thanks for setting this up.
Thank you, Johnny and Tom. Very nice.
Thank you, Tom.
Thank you.
All right, everybody go enjoy your Saturday.
Well, let's move on to our general discussion and open it up. Anybody have anything you want to talk about?
You want to turn off the recording so that you get a clean
Well, we'll do that a little later.
Okay, fine. I
Yeah.
was,
Mhm.
I'm gonna hang out if that's okay.
Yeah. Anybody? Does anybody want to talk about something you don't want recorded for posterity? Well, this is lively. Well, all right. If we're all out of things, I have something to show you. It's my latest generation in this plasma study I'm doing. It's a slide deck. So I guess you will be subjected to that. Or some of you might even be interested. I think it's one of the two most, well, now that we know about mitochondria, one of the three most interesting, exciting things we could do. So, okay, bear with me just a moment here. I will set it up. All righty, and I will share my screen. And I trust everyone can see what we're headed for. This is a real rich guy. That's the grave. This is the Grim Reaper standing between them. Grim Reaper is pointing to stacks and stacks of cash and jewelry and trophies and money. And this guy just telling him you can't take it with you. So, well, about that. This is what I'm going to do. This is self-explanatory. I'm working on it. And those of you, all of us here to a limited extent, very limited extent, we've done some of this, but we need to take it not from like here to here, a year or two, or maybe even 3 or 4, 5 or 10. What we're going to do is take it down to 20, 30, 40 years rejuvenation. So I'm Johnny Adams, and everyone here, we know each other. We've known each other 10, 15, 20 years, some of us, but my life for the last 4 decades is working on aging solutions and building skills and meeting people like all of you, and refining methods and building broad practical skills, etc. You can read the rest of this. I'll send this slide deck to you. Actually, it's up on the website as well. But here, hey, we're still there. Yeah, I don't know what
Where'd you
happened. I think
go?
I had a internet disconnection. I'm glad everyone else is still here. So let us resume. Bang. So, okay, let's take a look at the website if it'll let us. This is agingintervention.org. This is the stuff I'm working on. Kind of goes on and on. All right, enough of that. And let's go back to, this is a background summary. I've been working on this plasma arena since about 2015. We started off in the young plasma arena and then shifted to umbilical cord plasma. So, okay, let's go back to this. And the website has a lot of different interesting things on it, including a link to our YouTube channel. So, all right. These are some of the organizations I'm active in. Okay, so I've researched and done dozens of age reversal therapies, and one of the two therapies that, well, the one that of the two that are the very best, covering the full spectrum, of the two, the one that's available now, the best is therapeutic plasma replacement. And that is removing blood plasma from seniors and replacing with thoroughly screened plasma from donors in their early 20s. And there is a misconception that this is some kind of vampirism, stealing the youth away from young people. Actually, there is scientific research I can share with you that shows that this is actually good for the young person as well as the recipient. So it's good for the donor. And there are no ill effects of giving many plasma donations over the years. And I can show you a study that demonstrates that. And there are variations of this: therapeutic plasma exchange, remove plasma and replace with saline and albumin. And then also TPI, just an infusion without removal. So for me, TPE obtained the best objectively measured and subjective results of any age management therapy I've ever had. And I've had a lot. And my expectation is that old plasma out, young plasma in will be even better. The concept is based on something I think everyone here is familiar with. The circulatory systems of old and young mice were joined. The old mouse got the young blood, the young mouse got the old blood. The old mouse became biologically younger, the young mouse became older. This is called parabiosis. All of you, I think, are familiar with this. The same principle applies in a different way to humans. We're not going to sew people together, but we can do it in different ways. So plasma is the liquid part of blood, makes up about 55%. It contains thousands of components, and some of the main categories are extracellular vesicles or exosomes, or acellular nanoparticles, cytokines, growth factors, metabolites, nutrients, and the other things that you can quickly read on the screen here. Okay, a few of the thousands of components, and let's drill down a little bit to the extracellular vesicles or exosomes. There are many categories of these, so this is just a brief description, and we're researching and learning more about this all the time. Okay, a few more examples: cytokines. Some of them include interferons, tumor necrosis factors, chemokines, etc. Various kinds of growth factors, GDF11. Many of us are injecting GDF11. I did some just yesterday. And here is a list of some of the others that are, and many others of all these categories are contained in young plasma. Moving along, a few others. Hey, guess what? Mitochondria. Tom and I had a conversation, and he advised me that if you were to infuse young plasma from young donors, you would get mitochondria, and that would be a good thing.
Thank you, Johnny. Thank you for adding a slide for us.
Heck yeah. Hey, I'm a big fan.
Yeah, that's great.
And for years, in addition to plasma therapies, young plasma, and the other thing is that I'm working on, the Yamanaka OSK, both senolytics and mitochondria, repeat mitochondria, have been on the top of my list of things that are really important. And I've been waiting for you to come along, Tom. So I'm really glad that you're
We try to
you're
fit it all
working on
together. Absolutely.
Heck yeah. More examples of different kinds of things like NAD, CoQ10, and others here. miRNAs, different kinds of hormones, albumin. Albumin is a detoxifying and an antioxidant, detoxifying factor, and an antioxidant, and antibodies, stem cells, progenitor cells. So now here's one of my take-home points, why I'm kind of keen on this. This full range of complementary and balanced components found in plasma from young people would be far more, in my estimation, more effective than one molecule or several in combination. So this balance is consistent with the target we're aiming at, youth. And there's also evidence that these therapies, that young plasma assists in dementia and Alzheimer's and Parkinson's. So donors of the plasma will be thoroughly screened, and the plasma will be tested. They'd be tested for genetics, antigens, heavy metals, APOE, spike proteins. There's a big controversy about that going on. We are carefully considering just how carefully we're gonna test for spike proteins, and they'll be interviewed just eyeball to eyeball to and be asked some, well, important questions about things that impact the quality of the plasma, including lifestyle choices and some other attributes. So one therapy, I contend, is like a piccolo player. That's nice. Combinations can be like a string quartet. Isn't that sweet? Now, here's something interesting. Sometimes the wrong combinations are more like a loud, out of tune, intoxicated rock band. And I think it was Jeff who brought to my attention that there are our bodies go through anabolic and catabolic periods. And if we have therapies that do both, they're kind of button heads. So if you want a soundtrack for that, might sound like this. Pretty awful. So young plasma, we're going to test, and I say is like hundreds of symphonies and choruses in harmony. Here's the Ode to Joy. All righty, this is so much fun. Also, there's evidence that these therapies assist in dementia, Alzheimer's, and Parkinson's. I guess it kind of sounds like. Okay, some scientific published research studies, and here's a list of them. You can get and read these for yourself on the website. It's the first link. And here's another one. Reputable, I think these are Stanford and others, some more, some other ones. There are some critics of some of these studies and how they're conducted. You can read about that and decide for yourself. Some more research, some more research studies. Tony Weiss-Correa, well, he did a video on it and also did a webinar, and there's a video of that webinar. And also even Peter Diamandis is talking about therapeutic plasma exchange these days, and he mentions therapeutic plasma replacement. So, and I have some other references for you. They're less formal and include some testimonials and anecdotes, and on the publications or things I put up on the website, I usually I'm very clear to state things that we're in process of researching, and that this is a research study, but also the academic peer-reviewed research. But I have some other things, if anyone's really interested in this, I would share with you that are not academic and high-level peer-reviewed, but still, they contain a certain amount of validity. These are some team members. Dr. Parita is our medical director and study site physician, and Deborah O'Neill has 30 years experience in plasma and blood banks and labs. She's our operations director, highly experienced. This is expected to be a profitable business. There's big old stack of $1,000 bills, okay? And let me tell you, I have skin in this game. I've put a lot of my own money and something that's a lot more precious to me, my own time, into creating the IRB protocol, paying for the IRB, the Investigational Review Board, and getting their approval. This is an IRB approved clinical study. And I'm seeking a partnership with someone who's in it for biological age reversal, not only the money. In fact, if you're just in this for money, this won't work. That's, we wouldn't be working together. I could introduce you to some people who are very good at that. Fiona Miller at Quadrascope, for example. So although this is expected to be a profitable business, if you're like me and have a passion to be biologically young again, this, I say, is the best thing we can do right now. And there'd be a fair return on investment. So you tell me how you'd want it structured. We can work something out. But like me, that should be minor compared to the age reversal effects. Here's what I want out of it. Surprise, I'm not in it for the money. But I want unlimited use of the therapies at very minimal, basic hard costs. And being part of this would put you on my personal A list for other innovative age reversal treatments that some world-class scientists and I are working on, including a Yamanaka OSK therapy, which I believe I mentioned. If you want to know more about this, contact me privately. I need $220,000 maximum to get this going. I could probably do it on less, but I want to make sure I don't have to go back to the well, and I want to have room for expansion. So what you get out of it, unlimited use of the therapies at very minimal cost, some at no cost. And here's a fact. Aging is 100% fatal. Tick-tock. These, you see on the screen, are unacceptable. The one on the left, sick, living in a nursing home, dependent on others, paying all our monies to doctors and hospitals, and eventually the thing on the right, the big sleep. No, thank you. At least we'll put it off as long as we can. I call it open-ended lifespan. Healthy, of course, and making the world a better place, because that's an important component of this, but we can talk about that later. You can't take it with you. No, you can't. This guy tried. How'd that work out? Not so well. Okay, so my groups are working on advanced therapies for open-ended life. Healthy, of course, and making the world a better place. And you want to be a part of it? So here's what I'm going to do. Mark your calendar, 2049. It's my big 100th birthday party, and that's where the long-term planning will begin. We'll do the things we did when we were young. Okay, we'll dance in the sun, dance till sunrise. We'll learn a bunch of stuff. We'll, I don't know, jump off cliffs into water. There's actually water there. Okay, we'll run a race. We'll hurl a hammer. We'll have a meeting. That's all what we need, more meetings. Long-term planning. We'll have some laughter, joy, and fun. That baby gets me. All right. Okay, you all know how to get a hold of me. So any questions? I guess not.
Well, Johnny, you mentioned you moved to Florida. Whereabouts are you doing this now?
Boca Raton, but also we're looking at a presence in Coral Gables, which is just west of Miami. But hell, I'll go anywhere. Also, we're planning on doing this in California, but Florida is a lot friendlier. Plus, the Yamanaka OSK group is centered here, and we're doing some things in Bahamas. I went to the Heal Inc. conference in the Bahamas, and they're doing some innovative things there. And Jamaica and St. Kitts and Nevis are just a hop, skip, and a jump away. And things, we're able to do things offshore there. But then there's also Cabo. So that's a long answer to a short question. So was there more to that?
No, no.
Okay. Hey, some of you stayed. All right. All righty. Well, I'm going to turn the recording off. So that's it for now.